Hello. Can you please unmute yourself and confirm whether I am audible?
Audible.
Okay. Thank you. So let's start this meeting. Actually, I was on mute and I did not see. I had started it on exactly at 7 p.m. So no issue at all. So let's welcome you. Today is the third week on the topic protein structure and stability in the course Fundamentals of Protein Chemistry. Let me first wish you happy Raksha Bandhan and let's start this meeting. As I was discussing in the previous sessions also, that if you have missed the previous sessions, previous live sessions, you can find my recordings on the YouTube. The channel is NPTEL-PMRF TA NOC22-BT62, and here you will find both of the recordings, previous live sessions, which is on week 1 assignment practice and week 2 assignment practice. Other than that, you will also find the week 0 assignment practice. Okay, so let's start this meeting. So as we all know, we are now in week 3, and week 3 deals with the protein structure and stability. I hope my slides are visible and I am audible enough. So welcome. I am Karan Kumar, Primary Research Fellow at School of Energy Science and Engineering, IIT Guwahati. And today, uh, our concept that we have covered in week 3, you must have gone through the lectures that are uploaded into the in NPTEL portal, which is related to the protein folding and stability. Here, the concepts that you may have encountered as like protein folding, then protein denaturation and protein structure determination. So here in protein folding, you must have studied different models, factors, thermodynamics, stabilization interactions, which are the interactions that are responsible for stabilizing the protein folding and holding the structural integrity. Then you must have gone through the protein denaturations and the factors involved in unfolding and what kind of denaturants you use in the protein unfolding experiments. Then also, ma'am has discussed about the protein structural determination, and you can do this by using the techniques such as experimental techniques, where you use the solution NMR, X-ray crystallography techniques, and then you can also explore the computational methods such as ab initio threading method, then homology modeling method, and other techniques to predict the structure of the protein. Then the important keywords are listed in the right hand side where you must have encountered about you must have read about the molecular sapnone, the framework and nucleation model for protein folding, then molten globules and the protein folding funnels, and conformational entropy related to the thermodynamics of the protein folding and protein denaturation. So now coming to the main topic, which is on the protein folding. First, I would like to discuss some very fundamental questions and related to this topic. First, can you answer this question just to let me know whether the brief understanding is there or not about the topic, why we are studying the protein folding and what knowledge is imparts on a general. Can you unmute yourself and answer this question? Why you are studying the protein folding? Can somebody unmute? Hello. Can any one of you unmute yourself and answer this question? Why are we studying the protein folding? Any one of you, no problem. And your answer can be in any of the language, either Hindi or English, anything, because this is an interactive session. So I am making it as interactive as possible. So yeah, your question is why to study the protein folding and what knowledge it imparts? Ranjit, Amrita, anything? Okay, no problem. Let me answer this question to you. Like, because to know the function of protein, like if because all the repairing function and whatever function are happening in our body that are because of the proteins. Proteins are the functional part of our body, and they play a major role in metabolism, in repairing mechanism, in enzyme function. Like they are the enzymes, they are the structural integrity. They provide the like different mechanisms. They play a major role, and the function of the protein varies because of its structure. Okay, so and the structures we have studied, level of structure we have studied in the previous module, which were the primary structure, secondary structure, and tertiary structure. Then you also study the quaternary structure. Then what are the role of these proteins in our body? And to understand its function, you must know the how it is folded. Like the only the correct folded structure will function at its maximum. Like it can only be functional when it is correctly folded. So due to the structural functional relationship, we must study the protein folding procedure, and we must know how it is folded and how it is functional. And there was another experiment, very excellent experiment, which was carried out, which is Anfinson experimental findings. And these are very much celebrated in the structural biologist group. This was discussed by ma'am in her lecture. And this particular discussion is very much important. Will and as per the research perspective, because later in the discussion session, I will also teach you how you can study, how you can predict the structure using the computational biology technique. And I will also discuss some of the some of the tools which you can use to study the protein folding, and you to predict the three-dimensional structure, and how you will go further after getting the structure. What is the next procedure to know how certain drugs are inhibiting particular protein, and how drugs are how people are studying the protein drug interaction, and discovering new drug related target protein, and different techniques. So I will also give some discussion on that. So let me start the questions. Okay, so your first question is, which of the structural motifs are defined? Which of the structural motifs are defined correctly? And your options are coil, beta strands. In coil, beta strands are coiled together. Option B, the Greek key, 4 adjacent parallel strand. Option C, zinc finger is one or more zinc ions, Zn2 plus destabilizes the fold. And option D, beta alpha beta, 2 parallel strands connected by alpha helix. So any idea of what could be the correct answer for this question? Okay, ma'am has provided some of the important structures in her slide. She discussed the different motifs, because this question is related to the structural motif. So to remember that, you need to study the structure and their particular kind of configuration, how they are folded, and how, because to know the function of protein, you must know their structure. Okay, so some of these are self-explanatory. Here in coil, it is you can reduce that in coil there must be the involvement of alpha helix, where this statement states that the beta strands are coiled together, which is not correct. So option A is wrong. In Greek key motif, if you study, if you have remembered, then the Greek key motif are the four beta strands, and then they are joined. They are connected in a very particular fashion. So here it is given the four adjacent parallel beta strand, which is not correct because these are not in parallel. Okay, then if you are like examining these options, then you will find this option D is the correct explanation, like which is beta alpha beta. Here beta alpha beta, two parallel beta strands are connected by alpha helix. So your correct answer here in question 1 was D. Okay, so here in motif, two adjacent beta parallel strands are connected by an alpha helix from an C terminus of one strand to n terminus of another strand. The most protein structure that contain parallel beta seed are built up from the combination of beta alpha beta motifs. This is the extra information about it. Then your second question is, which of the following name is given to the molecular chaperone? So molecular chaperone. Okay, so molecular chaperones are the protein structure which helps in the protein folding, right? During the lecture, ma'am has discussed that the molecular chaperones are nothing but kind of heats of proteins. Okay, so your correct answer for the second question was option C, which is heats of proteins. Okay, here I have given some of the family of the proteins. Here the general structures are given, and in the right hand side you will find the energy landscape of protein folding. So here you can find the general structure of immunity protein 17, which is mostly the alpha helix connected through some of loops. Okay, here is sporulation protein UNV, then prokaryotic E2 family. The structure is given here. You will find the combination of beta strands and alpha helix. Then coordine. Then DNA-K related protein. DNA-K related proteins are involved in the DNA replication. So here these proteins are majorly composed of the alpha helix domain, right, and some kind of loop attached to it. Okay, then in RNA binding protein you will find mostly it is composed of the beta strands, combination of parallel and anti-parallel beta strands. Then spore coat protein, spore coat assembly of protein. Here this is the beta strands are formed in a kind of cylindrical pattern, and so similarly there are plenty of protein structures are given. So you can take a look of it, and also ma'am has discussed few of the general protein motif structure, and those are very important in terms of like studying the while you are studying for your comparative exam such as GATE and other NET. So there the direct question are being asked about the protein to identify the protein motif. Okay, so it's better if you remember some of the common motifs such as Greek motif, then alpha beta barrel, and different motifs that ma'am has given in her slide. So here in this energy landscape where the protein is one dimensional protein is getting folded, and with the help of chaperone they are getting folded in correct manner. So here when intermolecular contacts are arising, as it is given here intramolecular contacts. So intramolecular contacts, as the name suggests, it will prevail in the same protein structure, like within a structure. Okay, so these are helpful while when the protein is getting folded from one-dimensional to three-dimensional or in tertiary structure, where first the protein is folded into its probable secondary structure, and then the hydrophobic hydrophobic collapse happen, and then you find out the like in your native structure, in the native state, which is the correct protein folding, you will find the energy will lower. Okay, so here in case of intermolecular context, in intermolecular context, you will find that in the when two subunits come closer and then they make a globular or some kind of seed patterns structure. Here the amyloid fibrils and amorphous aggregates. So these are kind of, this is the protein energy landscape where the protein is being folded in correct manner. So your next question is which option is incorrect regarding the molten globule state of protein? Okay, so molten globule state of protein, that these terms appear when you study the protein folding model. So uh, here your correct answer will be for option 3. The molten globule state of the protein is a is not a denatured state, because so the correct option is protein. Correct answer for question 3 is A. Okay, molten globules are collapsed state, which is correct. Then presence of native content of secondary structure, which is also correct. Then the more compact state, which is also correct. So the incorrect option among these four is denatured state. So your correct answer is A. Now in the next question, the residue that tends to cluster in the interior of protein. So as we all know, due to the intrinsic nature of the protein, hydrophobic amino acid residues tend to go into the core of the protein because of the hydrophobic collapse. So here, if you correctly find out the amino acids which are hydrophobic in nature, so the correct option will be here: phenylalanine, leucine, and isoleucine. So the correct option for fourth question is option A. And in my week 1 discussion, I had given the uh, technique to remember the hydrophobic amino acid residues, and those by that you can recall the phobia of Wamp curve. So phobia of a Wamp curve represents the hydrophobic side chain. Here the correct option is phenylalanine, leucine, and isoleucine. And your next question is formation of amyloid beta peptides occurs in which kind of disease condition? So you may recall that in the Alzheimer disease, these amyloid beta peptides are aggregated and they tend to form the Alzheimer disease. Okay, Alzheimer disease arises due to the accumulation of amyloid beta peptides. So correct option is B. Now coming to the next question, the reactants are more abundant at equilibrium. Okay, so this question is related to the protein thermodynamics, the folding thermodynamics involved in the protein folding. So here K represents the equilibrium constant, and delta G is the change in initial Gibbs free energy. Okay, so you need to choose the correct option for this. The reactants are more abundant at equilibrium because of which option? So here your correct answer is option number B, which is K less than 1 and delta G greater than 0. So this is the this is more abundant at equilibrium, because the relation between the standard free energy change and the equilibrium constant is when you are at the equilibrium, then delta G will be equal to R minus RT ln K. Okay, where the K is equilibrium constant and it is the ratio of unbounded form, unfolded form, and native form, which is the folded form. So at the equilibrium, both of these will be equal. So ln K will be 0. Okay. So when K is less than 1 and delta G is greater than 0, the reactant are more abundant at equilibrium. So your correct option is option number B. Then in your question 6, which is related to the change in free energy of reaction A to B is minus 70 kilojoule per mole under standard condition, then the reaction. Your options are will never reach equilibrium. Option B will not proceed simultaneously. Option C will proceed at rapid rate, and option D will proceed from left to right spontaneously. So can anyone of you attempt this question? Okay, if you want to attempt this question, please unmute yourself and answer this question. Anyone? Anyone would like to attempt this question? Question is, if change of free energy of the reaction A to B is minus 70 kilojoule per mole under standard condition, then the reaction, what will be the state of the reaction? It is not a numerical question, but it is a kind of imperative question. Yeah, anybody want to answer this question? What could be the correct answer for this question? Okay, let me answer this question. Your correct answer for this question is option D. Why? Why? Because here the delta G is negative, right? So if delta G is negative, that means the reaction will be spontaneous, right? The condition for spontaneity of reaction is delta G less than 0, right? Change in Gibbs free energy should be 0. Here in the question it is given that your free energy of the reaction is minus 70 kilojoule per mole in the right direction, right? So the reaction will proceed from left to right. That means the product will be formed spontaneously because the Gibbs free energy is minus 70 kilojoule per mole. Your next question is, X-ray are produced by interaction of accelerated. Which is the correct option for this question? Your option A is because of accelerated atoms. Option B is protons. Option C is electrons. Option D is neutrons. What is the correct option? What is the correct answer for this question? X-ray are produced by interaction of accelerated. Which subunit of the atom? Whether is it atoms, protons, electrons, or neutrons? Your correct answer is electrons, because X-rays are produced by the interaction of accelerated electrons with tungsten nuclei within the tube anode. Okay, how these are produced? So when a high beam X-ray, if a high beam of energy intensity of the light is bombarded on the outer shell of the nuclei, then the electrons in the outer shell, they will absorb this light if it is suitable enough to exert the electron from this, excite the electrons from this state to another level of the another cell. Then these electrons will absorb light and they will transit from one state to another state. Okay. Then during this process, they will release a kind of energy which is in the X-ray region. Okay, this is called the X-ray emission. Okay, so X-rays are produced by interaction of accelerated electrons with tungsten nuclei within the tube anode. Your next question is, to study the protein stability, urea is used for which purpose? During the protein stability study, why urea is used? Your option A is to refold the protein. Option B is to unfold the protein. Option C is to make the covalent bonds with protein. Or option D, to increase the conformational stability of the protein. What is the correct answer for this question? This was taught by, I mean, lecture 13, I guess. Yes. 13 or 14. 14. So why urea is used in protein stability studies? If you have correctly, yes.
Option B, to unfold protein.
Good, very good. So the correct option is to unfold the protein, because urea सबको mail कर दो. Yes. कल का yes sir. Okay, so your correct option here is to unfold the protein, because urea is a denaturant and it is used to unfold the proteins. So now your next question is, in the folding reaction of the protein, which of the following does not occur? Your options are decrease in entropy, option B, increase in concentration of native state, option C, increase in free energy, option D, decrease in concentration of protein in native state. What could be the correct answer for this question? Please note, the question is related to the folding of proteins. During the folding of proteins, which of the following does not occur? So you need to remember the thermodynamics of protein to attempt this question. What happens during the folding of the protein? Can anyone of you unmute yourself and answer this question? You can try this question. Whether the answer is correct or wrong, that is not the we will not judge here. It's just to interact more and get the insight whether you have watched the lecture and understood the concepts. So what could be the correct answer for this question? Okay, so here, if you have remembered, the in folding of the reaction, in the folding of protein reaction, here the entropy of the protein, like here the correct answer for this question is option D. Okay, because here the question was which does not occur, right? So here you find that when the protein folding reaction is happening, so in that case the entropy is decreases. Entropy decreases during this folding reactions. Okay, so option A is correct. Like in the folding statement, option like decrease in entropy, that is correct. Then increase in the concentration of native state. Okay, that is also correct, because folding means the 1D structure will get into the final state will be the correct folding of the protein. So your native concentration, the concentration of native state will increase, right? So this is also a correct statement. Then increase in free energy. This is also usually seen because you must have remember that during during the protein folding, the entropy decreases because the correctly ordered water molecules that surround the protein, they must be like the hydrogen bonding between them must be disrupted, and the hydrophobic amino acid residues are getting buried into the structure, okay, because of the hydrophobic collapse. And also the protein tends to decrease its entropy and increase its free energy. So all three options are correct statement, while the concentration of the native protein does not decrease, because the folding itself means the protein will go into its native state. So the incorrect statement here was option D. So the correct answer for this is option D. Okay. Now coming to your next question, compare the protein stability in these denaturant. Your options are SO4 or SO4 2 minus, chlorine chloride ion, urea, and SCN minus ion. What can be the correct answer for this question? Can somebody attempt this question? Anybody want to attempt this question? All four are the denaturant. Okay. All four are the denaturant for a protein. So your correct answer here is the strongest one is here is SO4 2 minus, because it is radical ion, right? So it will denature it will rupture the protein more faster. Okay. Then second most among the denaturants are urea, right? Then chlorine, and SCN is the least in like protein stability study. Okay, so your correct option for this question was option C. Okay, your next question is, which type of energy landscape is shown here? Options are the idealized smooth funnel, option B the rug funnel, option C golf course landscape, and option D the moat funnel. Ma'am had discussed many of the different landscape model where she has given this kind of example. So can somebody correctly identify which kind of energy landscape it is? Anyone? Your correct answer is golf course landscape. Okay, so the question, the correct answer is C. Then your next question is, which of the following is correct about NMR? NMR. Which of the following is correct about NMR? Option A, it stands for nuclear magnetic resonance. Then a nuclear magnetic resolution. Option B is nuclear magnetic spectroscopy. NMR spectroscopy observes spinning patterns of nuclei in the electric field. Option C, NMR spectroscopy observes spinning patterns of atomic nuclei in oscillating magnetic field. Or option D, NMR spectroscopy observes spinning patterns of atomic nuclei in rotating magnetic field. What is the correct answer for this? Ma'am will discuss this in the later module, but because she has taken this NMR to show you how this NMR technique can be used to study the protein structure, because there are two techniques using which you can study the correct folding, or you can which are the X-ray crystallography and solution NMR. So for including that to include that, I have put this question. The correct answer here was option C. The NMR spectroscopy observes spinning pattern of atomic nuclei in oscillating magnetic field. So the correct option is option C. This is incorrect because NMR stands for nuclear magnetic resonance, not resolution. Okay, so option A is wrong. Then NMR spectroscopy observes spinning patterns of atomic nuclei in weak oscillating magnetic field and produce an electromagnetic signal with a frequency characteristic of magnetic field at the nucleus. Okay, so your correct answer was option C, which is NMR spectroscopy observes spinning patterns of atomic nuclei in oscillating magnetic field. Then this question was also taken in the previous discussion session. So can someone of you recall this question and answer this? And dash is found by anti-parallel beta sheet where the first and last strands are connected via hydrogen bonding. Your options are helix turn helix, option B is Roseman fold, option C beta barrel, option D alpha domain. This was also discussed during the current module by ma'am, where she was showing the different motifs. So can somebody attempt this question? What is the correct answer for this? Anyone? Okay, so let me answer this. The correct answer here is beta barrel, because beta barrel, the structure of beta barrel are such that the alpha beta strands are wrapped around in such a manner that they form a barrel kind of structure, and these are the membrane protein. Okay, they form a cylindrical type of structure which is composed of the beta strands, and the examples you can see them in the transmembrane porine proteins. Okay, so correct answer is C. Now, which of the following is best describe a characteristic of a protein motif? Which of the following best describe a characteristic of a protein motif? This question was also taken in the previous discussion session. And also you have studied this in the current module. This was specifically related to the protein motif. Okay, your correct answer is a commonly occurring arrangement of the made up of multiple secondary structures. Okay, a protein motif or super secondary structure is defined as the arrangement of the secondary structures within a protein, and it is commonly occurring enough to have the identified structure. As example, the beta alpha beta low. While the arrangement is made up of the secondary structure, the overall motif itself can be considered as a super secondary or possibly even tertiary. Okay, though its components are secondary structure, motifs do not necessarily have the defined function across the different proteins, and protein domain in the other hand do. Okay, so the correct option for this was option B, a commonly occurring arrangement made up of multiple secondary structures. Next question is, if delta G is greater than 0 for the protein unfolding reaction, then what is the correct state for that particular protein? The unfolded state options are unfolded state is more stable. Option B, folded state is more stable. Option C, unfolded and folded both states are stable. Option D, unfolded and folded both its states are unstable. What is the correct answer for this question? Okay. As we all know that at the equilibrium, when at the equilibrium of this state where you have the native state and unfolded state. Okay, during the unfolding reaction means the unfolded state will be your final thing. So unfolded state will be on the right hand side and the native state will be in the left hand side. So the equilibrium constant for this reaction will be the ratio of the concentration of unfolded state and native state. Okay, so the difference of the Gibbs free energy of the unfolded state and native states are delta G minus RT ln K. Okay, so for a positive delta Z, that indicates the native state is more stable, because the delta G, if delta G is negative, delta G is positive, what it means? It means minus RT ln K is also positive. Okay, minus RT ln K. Minus RT ln K is also positive. Since the negative sign is here, which means the RT ln K must be negative. So R is constant. R is a constant. T is temperature, that cannot be negative. So ln K must be negative, right? So ln K is negative only when in the state. So now can somebody answer what can be the correct answer for this question number 13? So here the folded state will be more stable because ln K is negative. Ln K is negative means this ratio should be negative, right? Or wrong? Folded state here, the ln K is the where K is the ratio of unbounded and bounded state. So delta G is negative, delta E is positive. That means ln K is negative. Ln K is negative. It means the K should be less than 0. And if K is less than 0, then ratio of U by N is less than 0. That means the N will be more in folded state. Okay, so folded state is more stable. Now, if the changes, and you can also find, you can also understand this question by like if delta G is positive, that means the reaction will not be the spontaneous, right? So it will not proceed in the forward direction. Therefore, the native state will be more stable, and you will find more of the native state structures. So native state correspond to folded state. So correct answer is B. Now, if the change is free energy is 0 for a reaction, then what could be the correct answer for this question? So do you remember the equation for delta G? If the change is free energy is 0 for a reaction, then what will be the correct answer? Delta H denotes for enthalpy. So delta H is equal to delta T delta S, or delta H is equal to minus T delta S. Delta S is equal to T delta H, or delta S equal to minus T delta H. What is the correct answer for this question? So as you all know, the equation is delta G is equal to delta H minus T delta S, right? So if delta G is positive, then delta H minus T delta S is equal to 0. Then delta H will be equal to T delta S. So your correct answer is A. Okay. So at the midpoint of temperature transition curve of a protein folding. So if you remember the first description where I was showing you the energy landscape. So the question is related to that only. At the midpoint of temperature transition curve for protein folding, what will be the correct statement? The concentration of native structure will be equal to unfolded state, or the concentration of native proteins will be greater than the unfolded, or the native protein state will be the less than unfolded state, or the native will be 0. So your option is at the midpoint of temperature transition curve. Your correct answer here is, in the energy landscape, if you see that at the mid temperature, at the midpoint of transition state, both of the state will be equal. Okay, so your correct answer is option A. Okay, your native and concentration of native state and unfolded state both will be equal. Now, next question is, myosin and keratin are alpha keratin are example of which kind of motif? Which motif describe the myosin and alpha keratin? Your options are coil. Option B is beta meander. Option C is helix turn helix, or Greek key. Okay, so this was discussed in the lectures of the while ma'am was discussing about the protein motifs. So you must remember that myosin and keratin are type of coil structure. So in these cases, alpha helix are prevalent and the alpha helical structures are wrapped around and they form the myosin and keratin structure. Okay, and these are called the coil motif. So correct answer is A. Your question number 17 is, interaction of helix turn helix motif with DNA is. What is the correct statement for this question? Option A is C-terminal helix fits into the minor group of DNA. Option B, the N-terminal helix fits into the major group of DNA. Option C, C-terminal helix fits into the major group of DNA. Or option D, the N-terminal helix fits into the minor group of DNA. What is the correct statement for this? Okay, so if you will study the molecular biology, in molecular biology, the DNA is the helix turn helix are mostly interacting with DNA. Okay, while in the DNA replication cases, the most of the motifs that interact with DNA are type of helix turn helix, and the correct statement of this question was option C. Here the most DNA binding motifs of alpha helix turn alpha helix motif interact with the side chain with the nucleotides in a sequence specific manner, and C-terminal helix fits into the major group of the DNA. So your correct answer is C-terminal helix fits into the major group of DNA. Your last question for this discussion is, which of the option is incorrect regarding the molten globule state of the protein? This we discussed in the first discussion slide where I was showing the where I was showing the energy landscape of a protein folding. So the molten globule state itself, it defines that your correct answer for this is the denatured state, because here the question was to identify the incorrect statement. Okay, regarding molten globule state, this appears during the protein folding. Okay, so protein folding means the structure will be in the collapse state. So option B is correct statement. More compact state, this is also a correct statement. Then option D, the presence of native-like content of the secondary structure, this is also a correct statement. The incorrect statement of this question was is the denatured state. Okay, so molten globule, the explanation is molten globule state protein is partially folded protein state, but not the complete denatured state. Okay, so Option A is incorrect statement in here. Now let me tell you the different. Ma'am has discussed the different techniques to identify the protein structure, structural folding patterns. So there you studied the two category. One is experimental technique. You can identify the protein folding structure using the experimental techniques. Another one is using the computational techniques. Okay, so in computational technique and in experimental techniques, the major difference is the time. Okay, you can get the correct structure of a protein by using NMR solution NMR and X-ray crystallography, X-ray diffraction pattern that I discussed in the previous slide, previous discussion session of the week 2. Here I will discuss about the NMR solution technique. Here, how you perform a NMR solution NMR technique is like first you isolate a protein. Okay, isolation techniques that I had discussed in the last discussion session of week 2. So you isolate the protein and then you purify it using the column chromatography technique, and then you put into a NMR tubes. Okay, here in NMR tubes you put some solutions and label the protein structure, and then this is the NMR instrument. Here you can create the 3D or 4D NMR patterns. Okay, and then you check the signal assignments of each peak. This will give the peak, and this is signal assignment you can perform using by different databases, or you have the libraries like here for different targets. If you collect the data for NMR, then using the fragment library, you can correspond the particular NMR spectra to its structure. Okay, and there you will see the chemical shifts and the major differences and different patterns in the NMR like NMR data, which will give you the peaks. Okay, and then you identify them using the library, and then you simulate annealing based on the techniques. Like then you simulate annealing of the various protein 3D or 4D NMR data, and then you identify how this protein is folded. Okay, using the different dihedral angle data or the protein distance. Then you impose certain restraint onto the structure, and then finally you validate the structure using the Ramachandran plot, because you know different if the the secondary structures are not modeled properly, then there will be a clashes between the groups, and Ramachandran plot suggests the which kind of rotation or which kind of dihedral angles are possible in a folded state of the protein based on the allowed region and disallowed region. Then you validate the precision and accuracy of your annealed structure, and finally by this iteration you follow the iteration many times, and then finally you get to know what is the correct folding stage of a particular protein. Once you know the correct folded structure of a protein, then you can go and explore its functional property. Okay, so here the another technique which is related to the computational biology. Previously I have shown you the experimental techniques, how you can find out using the experimental techniques. Now this technique, like in computational biology, what you do, you first obtain a protein sequence from the various databases. Okay, so ma'am has touched upon this computational technique in very briefly, but here I would like to give you a detailed explanation of it, because if some of you are interested in knowing the computational biology techniques to identify the protein structure or the correct folding of the structure, then this is very important in your research purpose, like to identify the knowledge based, like if you want to understand the knowledge based search of the protein, like to identify the docking and to identify the drug to propose for the drug repurposing, then to understand the correct target of a protein, to identify the vaccines, to discover the vaccines. These all are related to the protein structural identification. Okay, so here if you know the correct state, then you can identify the correct function of its of the protein. So this you can do by the computational biology technique also. And these, why computational biology techniques? Because they are less time consuming. And if you have the protein sequence, the basic necessary thing to model a structure is to know the protein sequence. It means the first primary structure of the protein, because ma'am has also repeated many times, the information related to the protein folding is deep encrypted in the primary structure of the protein itself. Like in the protein sequence itself, the information is hidden, like how the protein will fold. Okay, so to know that, you need to have the protein sequences, then you will do the database searching. Database searching are related to know which kind of the category of this protein is, like in which category they fall, whether it is a kind of membrane protein, whether it is kind of a globular protein or some other kind of. There are many database, like ma'am has discussed two, which is scope, scope, and cath domain. Okay, so I will also show you how which all are other database for the protein search. Then in the next step, you go for the multiple sequence alignment. Multiple sequence alignment is to identify whether whether the correct pattern, like the homologue, is already existed in the protein data bank. Protein data bank is the repository for 3D structures of the protein, which were solved using the experimental techniques such as X-ray crystallography or NMR solution technique. Okay, so then you go for the multiple sequence alignment and you find out, you try to find out whether the homologue is existing or not. So if homologue is existing in the protein data bank, then you go for the homology modeling of the protein. For that, you will need a template, and that template you need to select based on the multiple sequence alignment. Okay, if you find out the template, then you go for the homology modeling technique, and then you finally obtain a 3D model. If you don't find a homologous of your query sequence, then you go for the ab initio method. In the ab initio method, first you need to identify what all kind of secondary structures are available in your sequence. That you can do using the secondary structure prediction tools, which are like for the secondary structure prediction tool, you can have the tools such as RaptorX, FoldX, EasyPred. I will show you these all tools. Let me first discuss this the root or like the main procedure of obtaining a 3D model. Okay, then if you correctly find out which all the secondary structure are possible in your protein sequence, then you go for the fold recognition. Then if fold is recognized, means the protein predicted fold is available, and you identify the fold, and then you find out the template for it, and then doing the sequence structure alignment, you can again go for the homology modeling, and similarly you will end up with a 3D structure of the protein. If you don't find a fold, the correct fold, then you go for the ab initio structure prediction method. Okay, so right here in this procedure, you will find you will need a different tools or database to correctly find out the to obtain the 3D model of your protein query sequence. Okay, so you will need first you need to acquire the sequence, and let me share my okay, so here using the multiple sequence alignment, then you go for the homology modeling, and homology modeling you can perform using the tool which I will suggest you to use, the modeler. Okay, modeler is a program. You can download this, and then you can perform using a like command. It is a command line based tool. Okay, and then other than that, you can also perform the homology modeling using the the list of tools are given here. You can use the intfold, then RaptorX, then Fire2, then Modeler, then Robeta. So these all are for the homology modeling. And if you do not find the template for your protein, then you go for the thread and fold recognition. And for ab initio structure prediction, you can go for these tools. Okay. Now, if you have find out the homology modeling, then you need to correctly find out the models, like based on the parameters, and then you go for, then you finally get the protein model of it, like protein model of your query sequence. Then what you need to do, you need to check with validate the model, then that you can do using the tool which is Ramachandran. You can do the Ramachandran plot analysis. Ramachandran plot. This you can plot using the tool is this one. Okay, here the description of Ramachandran plot, we have already studied about it. Then you can also perform the analysis using the tool such as a protein structure validation. Okay, you can go to this website, and then you can perform using the mol probability, ProAssay, Watt check. These all you can use to validate the structure. If you have validated, then you can finally go for the molecular docking. And in using molecular docking techniques, you can identify which are the target for your drug, and other information you can get and then finally you can propose the drug. So if you want to get a detailed lecture about the molecular docking, then you can comment on the my YouTube channel, and then you can, if I get the enough responses from you that if many of you wanted to have this molecular docking lesson and homology modeling techniques, then I will take a separate lecture on that, which will not be related to this module, but that will help you in finally to get the computational biology techniques, how these tools are used in the research. So based on that, now finally, I thank you all for patience listening, and I will upload this lecture on YouTube channel, and then you can you can rewatch it on my YouTube channel. Okay. Are there any questions?
No, sir.
Okay. Thank you so much for joining the session, and I will end the session now. Thank you. Happy learning.